Well, hello, dear colleagues, dear friends, dear partners, hopefully, the future partners, and everybody who's interested in functional precision medicine. I'm very happy to meet you here. I'm very happy to see you here, because this is the second year I am organizing this functional precision medicine session, and the reason why is because I am seeing that for many, many people, the word combination precision medicine associates only with the genomics, like the NGS and so on, and nothing else. So here with this session, I want to show that there is something outside of the genomics, and I'm very happy for the speakers this year. And I'm very happy also for my co-chairs. So I will introduce myself. My name is Inese Cakstina Dzerve. I'm from Riga Stradins University, and I am basically the cell and molecular biologist, and I have very wonderful co-chair.
Yeah, thanks, Inese. So I'm Caroline Heckman. I'm a research director from the Institute of Molecular Medicine, Finland, and the University of Helsinki, and this is my second time at this meeting and in the functional precision medicine session, so I'm really looking forward to today's speakers, and I think Inese.
Yes, and the way how we will do today is we will invite the speakers. There will be 15-minute speech, plus minus 1 minute. I will raise my hand. Yes, and afterwards we're going to have like 5 minutes for the fast, like urgent questions that arise. And then at the end of this session, we have four speakers. At the end, we're going to do the panel discussion. So after you finish the talk, you can go back in a hall, because then you can see the presentations from the others. And at the end, I'm going to invite you back on the stage. So I am super happy to invite our first speaker, Diana Azzam. She's from Florida University. She's associate professor, and she's going to be talking about using functional precision medicine to guide cancer treatments. We can applaud.
Good afternoon, everyone. It's a pleasure to be here. Obviously, I want to start by thanking the organizers, especially Inese, for the invitation to speak today. I'm very excited to tell you about functional precision medicine. And really, the goal of my session is to give you the foundation principles of functional precision medicine. And hopefully, I can convince you that it is the result of major technological advances in functional drug testing that enabled us to integrate functional drug testing with genomics to impact patient care. I don't think this is... Oh, there it is. Okay, so my disclosure, I'm co-founder and scientific advisor of First Ascent Biomedical. So as I mentioned, really the focus today is, I hope by end of the session, you will get a good idea of how major technological advances and the growing list of FDA-approved drugs have enabled the bridging of genomics with functional drug testing as clinical decision support tools to impact patient care. So as you all know, precision medicine today commonly uses genomic profiling or DNA-only profiling to look for changes in the DNA of the tumor and to identify targeted drugs for each individual patient. What functional precision medicine is adding this additional component and integrating with genomic profiling, where we test hundreds of FDA-approved drugs directly on the patient's own living cancer cells to identify the right treatment for each individual patient. And that's called ex vivo drug sensitivity testing as well. Our study was featured in April on the cover of Nature Medicine, and what really this depicts is this gigantic maze which shows you the challenges that clinicians usually face when selecting treatments for their patients whose cancers are refractory, or whose cancers have returned, or whose cancers are rare. Is like entering this maze. And today, tools that include genomic profiling, ex vivo drug sensitivity testing, and artificial intelligence would provide an effective path forward for physicians and their patients to select the right treatment for each patient. So genomics precision medicine has provided a lot of impact or has benefited a lot of patients. But when it comes to pediatric cancers, there are unique challenges in using just genomics only precision medicine. First, because pediatric cancers have lower frequency of genetic mutations than adult cancers, and relatively few known driver mutations. So what happens if we can't find an actionable mutation? What do we do with those patients? Since then, a couple of precision medicine programs around the world started to incorporate functional drug testing, including INFORM in Germany, ZERO in Australia, Profile in Canada, and EITHER in Netherlands. Even then, we have very limited prospective trials that have looked at how functional drug testing can be implemented in the clinic. So my group and I, my group collaborated with Nicklaus Children's Hospital in Miami, Florida, a few years back to conduct a clinical study to determine if it's feasible to generate functional precision medicine data and return results back to a tumor board to direct the next line of therapy. So we performed this on relapsed refractory pediatric cancers, both solid and liquid cancers. And what we demonstrated that it was feasible to return FPM data to a molecular tumor board within a week and quick enough to direct the next line of therapy for these children. Since this was an observational study, it was up to the treating physician to decide if they wanted to use the FPM data to guide their next treatment regimen for the patient. So we ended up monitoring, or we ended up comparing clinical outcomes of patients who were guided by FPM, they were 6, and compared their clinical outcomes to those that were treated by physician choice. So what does the workflow entail? As I mentioned, we received samples from all types of tumors, both solid and liquid cancers, and we derived short-term cultures, as you can see here in the right bright field images. The cultures were short-term. They were a mix of 2D, 3D cultures that were semi-adherent and free-floating, as well as individual adherent cells. And within a few days, we tested a library of 120 FDA-approved drugs. That library included formulary drugs that are present at the Nicklaus Children's Hospital pharmacy. It included FDA-approved drugs that are in late-stage clinical trials for pediatric cancers, and it also included a list of non-cancer drugs or drugs that have been potentially investigated for repurposing in cancer treatments, such as statins and allergy medications. We tested each drug at 10 different doses, and we derived a drug sensitivity score. The higher the drug sensitivity score, the more effective the drug. And then we calculated IC50 of each drug, and the drug whose IC50 was lower than the Cmax was chosen. And Cmax is just the maximum concentration of the drug that can be achieved in the blood based on pharmacokinetic data. So as you can see here in the waterfall plot, the higher the DSS score, the more effective the drug. What we also did was we used the physician's experience in combining these drugs in order to basically go back and test combinations of drugs based on their own experience of toxicity. And that also helped the physician make decisions on which combination of drug was less toxic. In parallel, we had to validate our cultures. So we used a variety of different approaches, such as we validated pathology-defined markers using immunofluorescence. We validated genomic alterations using qRT-PCR. And then we also performed immune cell type deconvolution, or RNA sequencing, and identified that over 90% of our cultures are tumor cells, but what was surprising is we also had other immune cells and stromal cells within our cultures. So we believe that the heterogeneity that is present in our cultures resembles the original tumor from which we derived these cultures. And this is also to show you that we had this 10% of immune cells and stromal cells within our cultures, as you can see here. Now, in this study, we did not include immuno-oncology drugs, but based on this data, we're starting to add immuno-oncology drugs as well without having to co-culture these cultures with T cells. But this is work that we're doing right now in progress. So out of 24 patients samples that we received in the lab, we performed out of 24 patients, correct. We performed genomic profiling on 20 patients, and the genomic profiling was a pathway-focused panel. We used UCF 500, and we also performed drug sensitivity testing on 21 patients. When we compared the time it took to return drug sensitivity testing to genomic profiling, drug sensitivity testing results significantly outpaced the time it took to get the genomic profiling results. So the way we did this, since these are children that have progressive cancers, the molecular tumor board would meet and recommend treatment based on drug sensitivity testing first, and then if we found an alteration or if we found an alteration from the genomic profiling, then that matched drug would be modified and added to the treatment. Out of all the patients, using genomic profiling only, we identified treatments for 5% of patients. But when we included the drug sensitivity testing, we identified treatments for 100% of the patients. And panel E and F shows you all the QC results that we had to do to make sure that our assay passes quality control. As I mentioned, because this is an observational study, we compared clinical outcomes of patients patients that were guided by functional precision medicine by measuring or looking at overall best response. So patients that had partial response and better patients that were guided by FPM had better outcomes compared to those patients who were treated by physician choice, and their best overall response was at least partial response and higher, as you can see here in this plot. And what's very interesting, and I want you to pay attention to, is that each patient had their own treatment regimen, and many of these treatments were readily accessible chemotherapy drugs. And knowing that this is a pilot study and it's heterogeneous and small, we used a commonly used metric in precision oncology, which is basically we compared outcomes, or we used patients as their own controls. So we compared the progression-free survival of patients to their own previous regimen. And if the ratio is greater than 1.3, this means this is a positive outcome. And what we found was that patients whose treatments were guided by functional precision medicine, or 83% of these patients had at least an 8.5-fold improvement in that ratio compared to only 13% of patients that were treated by physician choice. So I know this is a pilot study, but I want to take a few minutes to give you an example of some of the patients on our study that we learned a lot from, and really to demonstrate the power of drug sensitivity testing and how we can combine that with genomic profiling. So this is patient 13, 5-year-old boy with a FLT3 mutation, with refractory AML, and has a FLT3 ITD mutation. This patient had already gone through chemotherapy, had already a FLT3 inhibitor, and did a bone marrow transplant. And then the patient relapsed, and that's when he got into our study. So when we performed drug sensitivity testing, the first thing we were able to do is we tested all the FLT3 inhibitors that are still out there, and we found the most selective and most effective FLT3 inhibitor that still worked on the patient. And then we tested 120 drugs, and we found a combination of drugs that was most effective for his treatment. So we found that 2 drug combinations were as effective as 3 drugs when we compared the DSS scores. And so the physician ended up deciding to treat that patient with 2 drugs versus 3. So we got rid of one that is toxic to the heart. And then what was very interesting was the doubling of these cells with steroids. So we found that these cells increased in number. His cancer cells doubled in number with steroids. And this was important information for the physician to not to give the patient steroids to suppress the immune system. So given all that information, the patient was guided by functional drug testing and achieved complete response in 33 days compared to 150 days in his previous regimen. And not only that, he went through another bone marrow transplant, and today the patient has been in remission for over 2 years, more than 3 times his first regimen. This patient here is a 7-year-old girl with metastatic rhabdomyosarcoma. Now, in this patient, what I want to show you here is that what drug sensitivity testing provided was that she was still responding to vincristine and irinotecan, which was used way when she was diagnosed. So if it wasn't for that drug sensitivity testing results, the doctor wouldn't have given her that same regimen again. So the drug sensitivity testing showed that this regimen is still effective. And as you can see here in the PET scan, when the patient, before going into our study, you can see the tumor grew, but when it was guided, we found that the tumor decreased, and this was partial response. The other patient I wanted to mention here is an osteosarcoma patient, 9-year-old girl. Osteosarcoma is an aggressive type of cancer in the bone. And you can see that the NCCN guidelines in the left show you the different drug regimens. And what I wanted to show you is that drug sensitivity testing results sometimes implicate drugs that are, as part of the NCCN guidelines, which is great. But if you look at the top most effective drug, this is idarubicin. This is, a topoisomerase inhibitor that is readily accessible and cheap, along with montelukast, which is an allergy medication. So if you compare the cost of the drugs, if you compare the type of drugs that FPM can implicate, it's way cheaper drugs than the drugs that was given just before the patient got into our study. So how are we advancing the workflow today? We are starting to integrate whole exome sequencing, RNA transcriptomics, and functional drug testing, along with artificial intelligence, in order for us to start identifying drug response biomarkers for patients. So think about this. As we build this data set, hopefully in a few years, we will start identifying drug response biomarkers and predicting individualized treatments for each individual patient. We received funding from the National Institute on Minority Health and Health Disparities because the population in Miami is largely minorities. And so what we found is that this group of patients respond differently to FDA-approved drugs than other ethnic groups. And so we're using AI now to identify biomarkers that may be specifically expressed in minority populations and along with specific FDA-approved drugs that they would respond to. This is an example. I'm going to skip that maybe because I don't have time. Exactly. See, I felt it. But I do want to say something, and I'm going to take maybe the last slide to go over this. So this is what we're optimizing now in our ongoing study that we have open on 65 relapse refractory pediatric cancers. So what we're starting to do is serial DST sampling. And this is a 5-year-old girl with metastatic osteosarcoma. So we got a sample from her primary tumor. We tested hundreds of drugs. We found everolimus, which is an mTOR inhibitor, to be effective. The patient was given everolimus, but then we got another sample from her lung, which had metastasized to the lung. And what we found was everolimus was ineffective in the sample from the lung. And so the patient did not respond to everolimus. And so we ended up testing hundreds and combinations of other drugs, and the patient was given these drugs to the right, as you can see here. And what happened is, what we were able to do, this is 2 years today, the patient is being managed by this. Let me just move to the next slide. Sorry. So this is just to show you that now what we can do is we can actually start getting these DST from different sites and identifying what would work for that patient. And you'll see that her treatment was just modified over time. And this patient, again, is today 2 years surviving this really aggressive type of cancer by just managing and changing her treatment based on drug sensitivity testing. So I'm sorry, one final slide. This is important because I think what we're starting to do is using DSS scores. I mentioned to you that we're starting to get rid of drugs that are unnecessary. And how are we doing that? By comparing DSS scores. So, for example, this is a patient that, sorry, not this one. Let me move to the next one. Here, this is a patient that had spindle cell carcinoma. What I want to show you is what's in blue. And what's in blue is just combinations of drugs versus single agent. If you look at the DSS scores, you'll see that cisplatin on its own has a DSS of 35. If you add vinorelbin to cisplatin, the DSS score doesn't change much, which tells you that it's not needed. But if you add gemcitabine, the DSS score goes up. If you add paclitaxel, DSS score goes down. So this has started to be very helpful for the molecular tumor board to really make decisions on combinations of drugs and drugs that are not needed for the patient. Okay, we also have a study on adult cancers. I'm not going to go into details, but we're starting to optimize this on fine needle biopsies and core needle biopsies, and that's important to broaden access to all patients. And I'm also trying to get this outside a research setting, so we're trying to make this a clinical test, and we have to go through CLIA, so we are establishing a Center for Advancing Personalized Cancer Treatments and trying to get this test CLIA certified so we can potentially launch large-scale prospective multi-center randomized clinical trials to better assess the clinical utility of functional precision medicine approaches. I'm going to skip this. I do want to say that one of the most important challenges that we have to overcome is physician hesitancy. So I'm hoping that physicians start familiarizing themselves with new approaches in oncology and new types of data that will help them influence clinical decision making. And there's an urgent need for the medical community to collaborate with functional precision medicine researchers like myself, like Inese, like Caroline, because I think together we will be able to provide you new tools and technologies that will help you make better informed decisions for your patients. So I hope that we all come together and work together to bring this to patients as soon as possible. Thank you so much.
Thank you, Diana. Please don't go away. Please don't go away, because we do have like 5 minutes for the questions, and I kindly ask the auditorium, the people, if you have some questions to Diana, you have to go to the microphone, please. Thank you.
Hello. Thank you for your presentation. I have a question regarding the RNA sequencing part. Do you plan to expand the resolution of the analysis by maybe including single-cell sequencing, or in order to assess the possibility that the tumor is heterogeneous and there may be some subpopulations of the tumor that can respond completely differently? Or is this problem already alleviated by functional testing?
So we've made a lot of advancement in how we grow patient-derived cells in the lab. And what you see here is we're establishing patient-derived cultures short-term, and we are showing that these cultures really are very similar to the tumor in which we derive them from. So I think this is a great model today that we didn't have 10 years ago. We are growing these cells and we're showing that they're very similar to what they're derived from, and I think we should take advantage of these models to use them to guide treatments in the clinic.
Okay, thank you.
You're welcome.
Thank you. Any other questions?
I could ask a question. So thank you, Diane, for that wonderful presentation, and congratulations on that work. So as you're moving forward and including immunotherapies in your platform, I was just wondering, in a research setting, how accessible are those drugs? I think, usually we're just getting our drugs from chemical vendors, but for immune-based therapies, I think this is more challenging.
That's a great question, how expensive they are too to buy them. So I have libraries of drugs that you can commercially get from different sources today, but if you want to test and buy the immunotherapy drugs, you go from $100 to $2,000 for one, a small volume of antibody. But I think it's important that we include them because they're, at least in the adult cancer feasibility study that I have, most of the doctors are now starting to, use immunotherapy for many of these refractory solid tumors, and we have to be able to try to, come up with ways to predict immunotherapy response. So I agree with you. I mean, the big problem here is accessibility to drugs. So because I didn't mention earlier, in the children or the childhood cancer study, it wasn't a problem because insurance companies covered cost of off-label use of most of the drugs because they're children, but that's not the case in the adult cancer study. So we're starting to just test drugs that are approved for that cancer type, just so we can give better options for patients, but this is definitely a challenge, and I hope we'll find a way to get these payers to start covering costs of drugs in an off-label indication. Thank you.
Okay, thank you. We probably have to go to the microphone. That's the last question, and then afterwards we'll move on, because we're going to have the panel discussion at the end as well.
Hello. Well, first of all, thank you again for the wonderful presentation. I wanted to ask, some of the drugs you've mentioned weren't, at least to my knowledge, an immediate candidate, like there was some allergy medicine.
Yeah.
How do you pick which ones to test? I mean, you cannot test all of the entire library of the drugs available to humankind, right?
By the way, we can. Today, with the acoustic dispensing that we have, that's what I wanted to tell everyone, that today we can test hundreds of drugs in nanoliters quickly. That's what we can do today that we couldn't do, years ago. So yes, you're right. It's best if we customize our library and test drugs that we basically can give to the patient. But I do want to make sure that everybody understands that we have technologies today that enable us to test hundreds of drugs.
Okay,
quickly.
This is incredible, but if we're talking about combinations of 2 or 3 drugs,
correct,
I'm guessing this explodes pretty quickly. So
for combinations,
yeah.
What we're starting, because you can't test all combinations, right? So we're starting to test combinations that are given together already, like part of NCCN guidelines, combinations that physicians are familiar with, because again, you don't want to give combinations, they're very hesitant to use combinations that have never been given together. So we're limiting our combination testing to those that have been already shown to be given together for now. But with AI, as we integrate AI, not only we want to be able to identify which drugs work, but we want to know why. And so we're slowly, hopefully, with time, as we generate this data set, we can start predicting better combinations through AI.
So the why, you mean explainability techniques?
Sorry?
The why the drugs work, you mean you're using explainability techniques in AI as well?
Yeah, because you're integrating whole exome sequencing, RNA transcriptomics, drug response, all of that together will tell you at some point which drugs work and why are they working, what's the mechanism behind them working, and what are the targets.
So actually you mentioned the keyword that I was trying to draw to. So you are using your AI system to actually select potential combinations to test, right?
Correct, not now, but as we generate the data set, because this data set doesn't exist today.
Well, there are quite a lot of data sets out there that you can use to train.
Maybe we can leave this one to the panel.
Yeah, let's
discussion.
talk more.
I'm sorry.
Let's talk
yeah.
more after.
Definitely.
And yeah, sorry,
Thank
we have
you.
to move on. Yes, thank you, Diana.
Okay, for our next speaker, I would like to welcome Dr. Triona ní Chonghaile from the Royal College of Surgeons in Ireland, and she will talk to us about multiple myeloma.
Thank you so much, and it's absolutely brilliant to be here in Riga. It's my first time in Latvia. It's nicely raining, so I feel very at home as an Irish woman. I'm the second Irish woman to talk today, so that's also highly represented. I'm going to talk to you a much more focused study about BCL2 dependence in multiple myeloma. So multiple myeloma hasn't been talked about, just a brief update. It's a disease of these plasma cells that produce antibodies. Here's a H&E stain of it. Our median age is around 69 years of age, so our patient population is quite elderly. And unfortunately, this type of leukemia is actually incurable. So we have really exciting treatments, and they work for a while, but unfortunately then the tumor relapses, and eventually the patient succumbs to the disease. So at the moment, our survival rate's about 60% for 5 years. So my lab works with a hematologist called Professor Siobhan Glavy, and our goal is to try and do these functional tests on the tumor samples. And we're taking a much more focused approach to Diana. We're taking 3 kind of functional assessments. 1, we're going to look at apoptotic sensitivity. 2, we're going to look at epigenetic state. And 3, we're trying to look at metabolism. Now, if anyone works with multiple myeloma, you know that you get a bone marrow biopsy, and you can have anywhere from 200,000 cells to 2 million cells. So our goal is to try and generate technologies that will work really well on low cell input. For the purpose of this talk, you'll have to invite me back to hear about the other 2. I'm just going to focus on apoptotic sensitivity, okay? This is a reminder, I know you're all probably familiar with it, but venetoclax is this super exciting BCL2 inhibitor. It's the first drug that inhibited protein-protein interaction. It binds to this big, huge pocket of BCL2, inhibiting its pro-survival function. So what happens is it releases these pro-death proteins. They'll activate BAX and BAC. We get a pore formed in our mitochondria, and we get release of cytochrome C that goes on and induces downstream cell death, apoptosis. So why do I care about the BCL2 proteins? Well, they drive survival, and we have these really exciting drugs now that can selectively target the individual BCL2 proteins. In particular, venetoclax is FDA approved for CLL and AML. We have a nice review, if anyone's interested, on venetoclax resistance mechanisms. Okay, so how do we profile sensitivity of the tumor samples to BCL2 inhibitors? 1, we actually just measure cell death. We add these BH3 mimetics to the cells, and we measure which BH3 mimetic induced the most cell death. And 2, we use this assay called BH3 profiling that was developed in Tony Latai's lab. I did my postdoc with him some time ago. There we add these peptides. They're like pro-death peptides. You can just add them to the cells. If a cell relies on BCL2 for survival, we get release of cytochrome C. So when we add the peptide, you'll get this big, large release of cytochrome C that we can measure based off the bad peptide. If a cell depends on BCL-XL, when we add in the HRK peptide, we get this cytochrome C release. And again, if it relies on MCL1 for survival, we have a peptide that binds selectively to MCL1. So we sort of have 2 assays we can use to determine sensitivity of cells to cell death. Okay, so I'm going to talk you through just this one patient who came in to Siobhan's clinic. She was 2-year history of multiple myeloma. They were refractory to bortezomib, proteasome inhibitor, 2nd-generation proteasome inhibitors, immunomodulatory agents, and her disease had become a plasma cell leukemia. So the disease was just completely prevalent in the blood, and it was so viscous, her blood, that her little blood vessels in her eye were damaged. So we got a bone marrow aspirate in the lab, and we did our testing where we looked at which BH3 mimetic the cells were most responsive to, and here we measured death using an X and PI. And we measure whether they're sensitive or resistant. This sample, we had lots of cells because it was a plasma cell leukemia, so we could also BH3 profile them. And this work was done by Lindsay Flanagan, who was a PhD student in my lab. So just for the sake of this, I'm not going to go through all the peptides, but when we add a venetoclax onto the profile, you can see it caused this huge response of mitochondrial potential loss. And when we treated the cells ex vivo, the most sensitive of the BH3 mimetics, as measured here, was the BCL2 inhibitor venetoclax. So we had this data, I think, within 16 hours after the sample arrived, but that sample went off and was cytogenetically tested, and it turned out this patient had a T1114 translocation. So multiple myeloma, it's known that patients who have this translocation have a tendency to rely on BCL2 for survival. So Siobhan started the patient on low-dose venetoclax, and in the study, we just published it as a case study because a plasma cell leukemia hadn't been treated with a single agent venetoclax. And the study, at the time of publication, I think we were only at maybe 50 days or something like that. Now we're out here, you can see that this patient on a single drug, their entire body was full of cancer, and the paraprotein was reduced greatly. And if you look at the viscosity of the blood, an indicator that loads of leukemic cells were killed off, you can see that that's greatly reduced. So this is one story of a patient who had this amazing response to venetoclax, and they had a really improved quality of life as well, because the disease was so prevalent in the blood, they had no energy, they had no very poor quality of life. So this translocation occurs in about 20% of multiple myeloma patients. But the goal of my lab was, well, how can we use venetoclax, because it's FDA approved, and bring it to a broader patient population? So to move it outside this T11-14. So what we did is, again, we took a really focused approach. We did an epigenetic screen where we took 2 cell lines that we knew did not rely on BCL2, so we profiled them and had done their mimetics. One relied on MCL1, and one was a mixture of BCL2 and XL. And we screened them, the 2 cell lines, with these series of epigenetic modifiers. Lindsay did this first as a viability, and then, because I'm a cell death person, I had to go back and do next and PI staining with the lead hits. And what you can see here is we've graphed the area under the curve of the drug alone, so that's in dark blue here. And then if we add in our venetoclax, we shift the IC50, and so that gives us a reduced area under the curve. And so we can plot all the drugs based on which ones actually enhanced the IC50 or greatly shifted the IC50. So as I mentioned, we did this in 2 cell lines. Really interesting, the 4 top hits were the same across the 2 cell lines. But excitingly for us, 5-azacitidine was one of the top hits. So for those of you who are familiar with leukemia, you know that 5-azacitidine is already FDA approved with venetoclax in AML, in particular for elderly patients. So how 5-azacitidine works is it inhibits DNA methyltransferase. Supposedly then we can reduce the methylation of promoters and drive expression of various tumor suppressor genes. Remarkably, venetoclax had never been assessed with 5-azacitidine in multiple myeloma. So we went to see, is this really a useful tool? Here is the IC50 of venetoclax. You see it does not kill these cell lines at all. The value is 14.2 micromolar. However, when you add 5-aza, you get a great shift in the IC50 value. If we look across a panel of myeloma cells, all quite heterogeneous, in dependencies, you see that we greatly shift the IC50. So just shown here on this axis is IC50 in nanomolar, and we do it even in T11-14 translocated cell lines. So that was exciting. Right, how is 5-azacitidine enhancing the response to venetoclax? So their next question. So we turned to a tool we have in the lab, BH3 profiling. This time we treated with our drug, 5-azacitidine. Then we added our peptides, and then we measured release of cytochrome C. And when we did this, we found, one, you can see there in the red bars, an increased response to venetoclax as we'd expect. But 2, we also found an increased response to an MCL1 selective peptide. So that what that told us is we've more pro-DEP proteins binding up MCL1. Maybe I can show it better here. We did an IP then, pull down MCL1, and we looked at interacting proteins. And indeed, when you add 5-azacitidine, you have more NOXA, which is a pro-DEP protein, and more BIM bound to MCL1. So our next question was, well, how is 5-azacitidine enhancing the expression of NOXA? And there's evidence in the literature that maybe NOXA can be induced by the integrated stress response. So just to introduce this pathway to you, we have various stresses that can come from unfolded proteins, double-stranded RNA, nutrient depletion. And what they do is they try and shut off translation. So we phosphorylate EIF2 alpha, and that will shut off protein translation, so reduce stress in the cell. So one of the inhibitors of this pathway is called ISRIB, and what we found was when we added 5-aza to our cells, you'll see an induction of NOXA. It's working as well as we'd expect. We don't see any DNMT1. The combination, we see an increase in NOXA. But if we add in an inhibitor that works here, we decrease the amount of this NOXA protein that's induced. So then it kind of we were like, how is Noxa getting induced by the integrated stress response? And we went back to the literature and had a little read. It doesn't want to work. Okay. So these papers that came out in 2015 showing that DNA demethylase inhibitors can reactivate these ancient endoviruses in our genome. And what that can do is it can upregulate double-stranded RNA. So we thought, well, is this what's happening? And is this potentially how Noxa is getting induced following 5-AZA? So if that was the case, there we go. If that was the case, a particularly important sensor for this stress would be PKR. So that can sense double-stranded RNA, which would be like a viral RNA in our cell, and it activates this response pathway. So what we did was we measured double-stranded RNA in our cells. Here's with venetoclax alone. Hopefully you can see this, it's kind of bright, but there are these green punctae at the bottom. When you add 5-azacitidine, you can see this double-stranded RNA, and when you add the combination, you can see even more double-stranded RNA. And that's graphed here by pixel count here for the amount of double-stranded RNA. So it looks like the combination is inducing slightly more double-stranded RNA than either of the two drugs alone. So remember that pathway, if double-stranded RNA was responsible for Noxa induction, if we deleted PKR, we should protect our cells against this combination treatment. So that's what we did next. We knocked down PKR, we silenced it using siRNA. Here we have venetoclax alone, you see no death. 5-AZA alone, we don't see any death, but the combination, we see a robust amount of cell death. And that's in the purple bar. And if we knock down PKR, you can see we're protecting against that cell death. So it does seem like PKR signaling is important for this combination treatment. Again, just to show that PKR is able to induce Noxa, one of the proteins downstream, if we knock down Noxa, we're also able to protect against this cell death. So hopefully what I've shown you in the molecular mechanism part is we did an epigenetic screen, we found 5-AZA could enhance BCL2 dependence. If we add in an inhibitor here that blocks the phosphorylation of EIF2 alpha, that can protect against cell death. If we knock down PKR, which is this sensor here, that can protect against cell death. And if we knock down Noxa down here, that can protect against cell death. And so what we have is this Noxa protein binding up MCL1, and it means that BCL2 then becomes more sensitive and more responsive to that venetoclax. Okay, so is this combination treatment in any way at all useful? So we went back to our patient samples, we treat them ex vivo straight away for 16 hours, and we measure cell death. So we do this blinded, but the post-analysis, we did divide them genetically, because you'll see why. So these are our non-T1114. This is the amount of death. You can see 5-AZA doesn't really do a huge amount to these cells alone. Here's venetoclax alone. It has an intermediate response. And then when we treat with the combination, you can see in 7 out of 9 patient samples, we greatly enhanced the cell death. As I mentioned, we didn't know at the time, but venetoclax is really, T1114 patient samples are really sensitive to venetoclax ex vivo. The cell death is up at about from 50 to 80%. And so even though venetoclax worked very well alone, we enhanced the cell death in 2 out of 3 of these samples. And we can actually just use ex vivo venetoclax nearly as a biomarker to discern which ones are T1114 and which ones are not T1114. So with that, hopefully I haven't raced through too fast, but hopefully I've shown you we can use precision medicine to identify which BCL2 protein a tumor is reliant on. This is very exciting for BCL2-dependent tumors. We had that one patient who had a 10-month remission, and really they wouldn't have lasted the weekend because they had so much disease in their blood. We performed an epigenetic modifier screen to try and induce BCL2 dependence. How can we broaden the use of venetoclax? We found that 5-azacitidine, an already FDA-approved combination, worked really well in multiple myeloma. I think we uncovered more of the mechanism behind how this combination works. We show that Noxa expression goes up. This expression is reliant on PKR, and that we can inhibit MCL1. And lastly, we showed, irrespective of cytogenetics, this combination worked quite well ex vivo in patient samples. And so hopefully it has the potential to expand the patient population that may receive venetoclax. So with that, I just have to say a huge thanks to the people in the lab. This study is mainly done by the PhD student. She's now going on to do a postdoc with Stephen Tate. And my collaborator, Professor Siobhan Glavy, who's the hematologist, and the funding. And thank you for your attention, and I'm happy to take any questions. Thanks.
Thank you, Triona. So, are there any questions from the audience? There's a microphone on the side if anybody would like to ask. I think in the meantime, I was wondering, so for that one case that you presented in the beginning, was that patient just treated with venetoclax alone, or was that a combination?
Alone, no, single agent, yeah.
Okay. So, and then in terms of uptake in a combination of venetoclax and azacitidine, this is, this is the normal combination for an AML patient, but for myeloma it would be quite unique.
Yes.
And I'm just wondering about maybe the clinical concern in not including a glucocorticoid like dexamethasone, which is very standard backbone for myeloma.
Thank you so much for asking this question. I didn't have time to put it in, but we looked at whether adding a steroid, and I'm so glad Diana, we'll have to chat about this later, would be of benefit, because it is routinely just added to all regimens in myeloma, so you just normally get a steroid. And so we actually did the work, which was, well, is it of any benefit? So we tested adding it to the cell lines, and it didn't enhance the cell killing of the tumor cells. But what we noted was that the T cells become remarkably sensitive to venetoclax if you add a steroid. So my previous memory of dexamethasone was it could switch BCL2 and XL in T cells. So we looked at that, and they became extremely sensitive. So we actually, in the paper that's just published, we suggest not adding a steroid, which is controversial in myeloma, but I think venetoclax hasn't worked as well as maybe anticipated in myeloma, because it just got a steroid. And no one has done the real key mechanistic work to see, is there a benefit of adding the steroid?
Yeah, that's really interesting, because the clinical trials, with testing venetoclax, it was in combination with a
It was in combination with a steroid, yeah. So it was bortezomib and a steroid. Yeah. And so there was 2 deaths, right? And that has made people very cautious about using venetoclax in multiple myeloma, even though it does have there's a particular subpopulation that are very sensitive to it. Because the way the trial was run, there was 2 patients who unfortunately passed away, and so now there's been a real, a real turnaround in where they use venetoclax in multiple myeloma. So even though we found this, actually getting into clinical practice, we have had discussions, but it's not as, so easy.
Okay. Is there no question? No other questions from the audience?
Thanks.
Yeah,
well, thank you, Triona.
Thank you. Yeah, thank you very much. And moving forward, I am inviting now our next speaker, Christian Regenbrecht. He's from Germany, University of Göttingen, or Bonn. He'll tell you more, but he will going to be talking about functional precision medicine. Is this a real life, or is just a fantasy?
So thank you for the invitation, and thank you. I think I already got in your head with the title, because I guess at least 50% of the people now think of Queen's Bohemian Rhapsody. After these 2 brilliant talks, it's a little bit difficult to add really new angles, but I try to get that. So let's get how I got into the functional precision medicine thing. I made a postdoc at the Max Planck for molecular genetics, and actually, like James Watson, we believed if we sequence enough DNA, enough cancer genomes, that will solve the cancer problem. But recent studies have shown that in average, only 3 to 8% of the people really benefit. And of course, this is averaging through many different tumor entities are benefiting from that. In 2017, I stumbled over this paper from Amy Brooks, and I think it's very crucial because it explains still today at the university level, mostly cancer is taught as you have a normal cell, it acquires a mutation, then you have the cancer phenotype. And if that was true, it would be very easy to study the mechanistic roles of various proteins in the pathways in the cancer setting, and from there get a drug therapy against those aberrant proteins that what we call today targeted therapies, target selective drugs, would lead to apoptosis. But we know now that the biological reality is much more complex, and with that we have, with the same genomic background, different cell types at the same time, which lead in consequence to more sensitive and more resistant cells. And I think the understanding that with the same genomic background, cells can behave differently was just rewarded with a Nobel Prize this year. So with this dynamics, we know that some cells undergo apoptosis and other cells are surviving, and in the paper they were called the stress-induced cancer stem cells. So what do I think are the challenges of precision oncology? Large clinical trials basically say nothing about the individual patient's response to a therapy. I already mentioned that cancer gene sequencing improves therapy for less than only 10% of patients, and the marketing gag of the targeted therapies is only working reliably for a few tumor types in many patients. So with this hypothesis of the surviving cells, this also explains why from treatment regimen to treatment regimen, the chances of a successful progression-free survival or overall survival are decreasing. So I think I don't have to go into this, but taking the point, we heard before that genomics is currently, in the general public discussion, the thing of personalized oncology. But I totally agree that this is just one piece of the puzzle, and we need to put the pieces together. What we are doing, we are focusing on the 3D cell culture models. And as you can see, these are integral parts. We have the models that we try to use to understand cancer, and we have the omics technologies that we need for the stratification and identification of potential biomarkers. So as I said, we focus around the 3D cell culture space. And just to bring into your minds, what is the, let's say, average patient journey? Patients, either by early detection or with a clinical diagnosis, turn up in the clinic. They receive neoadjuvant therapy, later eventually surgery with an adjuvant chemotherapy. Then they're in remission, there's a clinical recurrence, and there's systemic chemotherapies followed by additional systemic or targeted therapies. So this is the average patient journey. And with our models, we try to get into the idea of what it's doing. I think here in this group, and also to save some time, can quickly go over this. So this is a scanning electron microscopy of one of the colon organoids we use, and how good these models are. And I think this is crucial also in explaining to the oncologist what really the biology is. So in my experience, when talking to the oncologists, and you talk about cell culture, they think HeLa cells. So we need to get that out of their heads. So it's not HeLa cells, it's very patient-specific. In fact, what you see here is the immunohistochemistry of a colorectal cancer diagnosed by a pathologist, and here the derived tumor organoid from the same patient. And you don't have to be a pathologist to see how similar these patterns are. In fact, and I'm happy that Diana has spoken so much about sarcomas, according to WHO, there are 120 plus subtypes of sarcomas. And we have established a very good protocol to take sarcomas into culture. And this is 43 models that we blindly sent to a pathologist. And she was able, those in dark green, to confirm the original diagnosis only from the model. And in the light green, it was most likely it was, and she was correct for all the cases, There's just one outlier, this angiosarcoma, but when we looked at the history, is that between the original pathology and the piece of tissue that we have received, there were like 3 different chemotherapies, underscoring how much selective pressure is put on a tumor by treating it and changing its morphology. So our models are very different types. So we have from melanoma to non-small cell lung cancer, and you can see here also the phenotype is very distinct depending on the tumor type. So we use these models to bring this directly to the patient. And that is the point where I have, in addition to my academic career, had to set up a company, AAC Oncology, to go rogue. Universities are slow, at least in Germany. They take too long to make decisions. And to be swift, we founded a company. We originally planned to have this as a non-profit company, but unfortunately in Germany you don't receive any public funding if you're a non-profit company. So we had to make it a profit company, and we just added a non-profit club called Cancer Rebels to pay for the patients to get the method done. So basically it's a non-profit thing. So we start with the consultation with the patient and see if the procedure is suitable. Then we need to coordinate with the attending physician. And here's one of the big differences to Diana's approach. We only test, although we could also test hundreds of substances, we only test what the physician or the oncologist would be willing to give to this patient, due to the ethical problem that what would happen if we suggest something that might work in the cell culture and the oncologist would say, but you don't get it from me. So we don't want to break this relationship between oncologist and patients, and so we limit ourselves into what the physicians or the tumor board would recommend. So then we take care of the sample collection and take care of the shipment, testing, so to leave as few work to the oncologist. And basically, I think what we are doing for the community with that is we bring it into the broad field. We take it away from just the ivory tower of the university hospitals, spreading it to normal hospitals and even normal doctors' offices. So then we generate an individual report, and we meet again for a joint decision, which empowers and enables the patient also to participate in the decision-making process. That can be either, as I said, with the oncologist and patient, or as part of a tumor board. So we have put a lot of effort into making it as easy and feasible as possible. So we designed a box with those tubes. Everything is compliant to IATA standards. So interestingly, we have received now from over 30 countries specimen to do what we are doing. We have, as we have material even from Saudi Arabia, we need to find a cooling solution. So this is all the technicalities that make it the real life and not just a fantasy to get this done on a global scale and not just in some exclusive institutions. So the screening is very easy. We grow the organoids, make a single cell suspension, do an automated single cell seeding. While the cells recover from the procedure, we prepare compound dilution plates, put them on the assay plate, and the readout is basically the cell titer glow assay. And what I show here is some real data of a patient, and I will show you the entire patient journey and show to you the crucial key points. So basically, of course, we are looking in these dose escalation schemes for something that is going as much down as possible. You see here drug A, drug B, didn't work at all, even escalating it over the maximum plasma levels. Drug C had a bit of a response. Volvox was quite good. Cobimetinib also worked fine. The control, as expected, did nothing. And it's not only that I don't know how I can play the videos. No, it's not a touch screen. So actually you should see some videos here. Okay, can you start both in parallel? Thank you. You can see here the drug that did work and the drug that did not work. So in one video, the organoids grow. In the others, you see how they fall into apoptosis. And this, I think, very well illustrates that it's not just some biochemistry we are measuring, but it's real-life data. Okay, this is the actual patient journey of the patient I showed to you. It was a relatively young lady in her late 40s suffering from an appendix carcinoma, normally quite good to treat tumor, but she had a Ki-67 index of over 70%. She received surgery, got a port, got Folfox, capecitabine, went into remission, but almost a year later, there was a recurrence, there was a re-laparotomy. And interestingly, so we received a piece of tissue, the NCT in Heidelberg received a piece of tissue for sequencing, and another company received some tissue for xenotransplantation. So I think it's easy to learn. Xenotransplantation takes much too long. We are in a time frame competitive to the sequencing people. But what is the real big problem is this in the box. That was just discussion with insurance companies about off-label use, about covering the costs and so on. And as we identified this as one of the crucial points, we have now, we are very short of signing the contract with 1/3 of the German public insurance companies that they will in the future pay for this to shorten this crucial period of time before the patient can receive the treatment. So, as I said, these are the drugs we can test, a plethora of about 150 cancer drugs. The report that we generate and give to the oncologist is according to ESMO guidelines for precision medicine reports. Until 2 months ago, I didn't know such a thing exists. But we have page 1 is simple language, so even the patient understands it. So we enable the patient to understand and discuss with his doctor. Then we have an estimate what we would judge is a good working drug and not working drug. Then we put in the raw data and definitions of what we do and how we did it. So that is then given to them. And as I said, these are the points of entry where we can help patients. But when we do it on the clinical scale, the pathology can do whatever pathology does today to give the people optimum standard of care. But in parallel, we do the organoids, meet at the molecular tumor boards. Then we have the discussions. And you know, those people who have attended molecular tumor boards, you either end up with like no actionable mutation, one actionable mutation, but different drugs that you could test, or a couple of mutations, and people are fighting about which drug to test, and then we can easily go into a retest and make a ranking into the drugs that were discussed in the tumor board. As I said, we don't see ourselves as a for-profit company, so we have generated an entire ecosystem. We have a network of patient organizations we're working with. We organize expert lectures and so on and so on. And we even now try to build an online forum to bring patients and specialists together so that patients don't have to visit Dr. Google anymore. We have heard about that, so we have seen in multiple studies how well the organoid treatment is superior to physicians' choice. And last but not least, I want to show you this case study. So that was a patient in her early 50s, and he was diagnosed with a cancer of unknown primary. That was in October 2022. And I spoke to the oncologist, and the oncologist, he was Austrian. He was with a strong Austrian accent saying something like, do what you want to do. She will not survive to see Christmas anyway. So that was his response. Now, we did what we wanted to do, and it took us about 42 days to test drugs, and we found out all the drugs that showed some response were drugs that are administered with triple-negative breast cancer. So we reported that back, and they went into imaging. And they did a high-resolution imaging of the breast. They found the primary tumor, eventually. They could eradicate that by irradiation. And cutting the long story short, since summer last year, she is officially deemed cancer-free. So this person is back in her normal life, united with her family, from what was a less than 3 months perspective. Just a quick thing, we have heard about that. You can, and this is, I think, one of the big exploits of functional precision medicine, use this in several types of clinical studies. May that observational studies, may that be studies designed by a number needed to treat, to show superior, or single-arm open-label studies. And basically that gives us an entirely new way of practicing oncology, and I think it's one of the most disrupting technologies we have today. So not to overdraw my time too much, I thank you very much. I just need to skip to the one picture I wanted to show you. Now this is not an animation. This is scanning electron microscopy, and it's just like you take a picture, move the stage a bit, and move the picture, the stage again. So it took us about 16 hours for this 20-second video to take, but I think it's so fascinating I didn't want to spare it to you. Thank you for your interest, and hoping we have a great discussion.
Thank you very much. Any questions from the, yes, go to the, yeah. Mm-hmm.
Thank you for a very nice presentation. Just out of curiosity, what's your capacity for testing patients? What can you take for a week, for example?
So the capacity, let's put it like this. We have, actually I have two companies. One company called Self-Phenomics, you just saw in the final slide, is providing organoid screening service for the pharma industry. So therefore we have a team of 18 scientists that work the entire day on the organoids, and we can scale up easily to over 1,000 organoids per month. 1,000 patients, not organoids, but 1,000 patients per month. So we have the infrastructure in place, we have the trained personnel in place, but that was also the reason why we slowly stepped into the market, because what is worse than telling a patient we might have a technology available for you, but we are booked out for the next 2 months. So that didn't work. So this is now how we start making it broader and distributing it in Germany, starting with 30 plus insurance companies with cancer patients, solid cancer, pancreatic cancer, and sarcomas, so rare cancer and hard to treat cancers.
Thank you. Any other questions?
Thank you.
Thank you. I think we can leave for the panel discussion, and oh yeah. Thank you, Christian.
Thank you. Panel already?
All right, so now I'd like to welcome Inese, Cakstina, Dzerve to the podium from our local representative from Riga, Stradins University. And she's going to tell us about implementing functional precision medicine in Latvia.
Yes, thank you, Caroline. My time starts now. I thank you, everybody, for coming here again. So, as I like to call myself, I am a baby in this great society, because I'm very young for the functional precision medicine. And as we already, as the name already title says, first steps, and these are literally first steps. And my first step was by applying, by the chance to go to USA, and thinking like, because my husband has to go, and I thought like, what can I do for 1 year in USA? And then I had this suggestion to apply for the Fulbright and go to Boston, because in Boston there is a Dana-Farber Cancer Institute. So I did the research. There are approximately 127 cancer labs in Dana-Farber. So I selected 7. The first one was Tony's lab, Anthony Latai. Luckily, his name starts with A. And his lab was doing the work with the apoptosis and apoptotic priming. And that has been always the subject that I was always afraid of. And I thought like, okay, I'm going for 1 year sabbatical, so I have to do something challenging. And I was very lucky to get this Fulbright scholarship for the visiting researchers. And I went to USA for year 22-23, stayed there for 9 months and worked at the Tony's lab. And actually, I was working together with Patrick Bola, who is more in doing the functional precision medicine in solid tumors, and he was located in Harvard Medical School in Laboratory of System Pharmacology. But it has been like very, very beneficial because that was the place, and actually meeting Tony for the first time, I learned about the subject. I learned about the topic of the functional precision medicine, because just like many of us beforehand, I thought that precision medicine is just only the DNA sequencing, looking at the mutations, and choosing the treatment based on that. After coming back last year in June, I was very happy that PMNET organizers allowed me to do my initiative and organize the special session for the functional precision medicine last year, which I did, and I got 5 great speakers, including Tony Letai and Patrick Bola, and also Caroline Heckman, who's now my co-chair. And actually, this PMNET forum last year was so successful that we ended up writing 2 grants together with Caroline Heckman and other speakers, and not only other speakers, because other speakers were invited other people like Triona. So it has been like really great networking and really great outcome from that. In addition, I received also the national funding for my project finally, 5 years. And finally I got the lucky ticket, and I'll talk about it a little bit later. And as I said, yes, the outcome of this networking, right now we wrote 2 grants, but of course this is not the end. We are looking for the future collaboration and future possibilities. So we've been talking a lot already about this precision medicine, and I really, really like how Tony Letai describes it in very easy words. Just let's take the tumor cells, put the drugs on them, and see which drugs are killing them the most effectively. So this is like in 3 little parts, in 3 little phrases, like what's the functional precision medicine, yes? Instead of the sequencing and doing elaborate analysis, looking for the mutations, driver mutations, etc. Here we are looking mostly at the cell response to the drugs. And as you've seen already from the previous talks, there are different models how we can, what we can use, like in which way we can use the tumor cells. Diana showed the work where they were using the 2D, 3D cultures with the patient-derived cell lines, the short-term. Christian Regenbrecht showed like beautiful results from the patient-derived organoids, the 3D system. There is also a possibility to incorporate the microfluidic system that we do not look here today yet, but I know that Joan Montero is working on that, making the microfluidic system for such functional precision medicine tests. Of course, we have the patient-derived xenografts. As Christian very beautifully said, it takes forever to develop them, approximately 400 days. And of course, in no clinical setting, we have the time for that. However, it's a very good model for the research because we are seeing how we can apply it in a clinical establishment. But of course, more research is needed. As beautifully Triona showed, we can't rely we can't just lay on our success right now. We have to move on and understand these mechanisms, how the drugs are working more and more. And of course, the patient biopsies. This is actually what me and Patrick was working in when I was in USA, and actually particularly Patrick is trying to do the test like where we can trying to assess like what's the minimal amount of the patient's cells that we can use to for this drug sensitivity test for the BH3 profiling. And of course, these would be patient biopsies because we are looking forward to do the drug screening as soon as possible, because while for many cancers we can wait, there are some cancers that are very aggressive and for which this such kind of test would be, I think the time is of the essence in here. So various approaches, and of course various things that we can look in those ones, in those models. Mostly in a clinical setting, of course, the most easiest ones, let's put the drugs and see the cell viability. they survive or don't survive. And we can do that on almost all the models. My personal interest, of course, is more this like mitochondrial priming, because it gives like a more in-depth analysis and I think more information. When we are looking at patients and patient materials, we are usually talking about these two groups. We are talking about the solid tumors and the liquid cancers. Yes, I'm like my interest is also more in the solid tumors, but as we can see, actually the field starts with the liquid tumors, and it makes sense because it is much more easier to get the biopsies from the from these oncohematological patients and so. Also, when we are talking about the methods, and here I'm talking mostly like particularly for the functional precision methods, and I took the slide from the Institute of Molecular Medicine, Finland. People are, there are three main approaches. We can use the luminescence readout. Usually then after exposure to the drugs, we add the cell titer glow, which is luciferase-based assay. And then we read out the luminescence, yes, in luminometers. Another one approach is very useful for the liquid cancers. Of course, it's the flow cytometry, which gives the, of course, cell viability readout, yes, but it gives us information about which cell populations are targeted by the drugs. And the third one is the image-based readout, which is the BH3 profiling, and not only, of course, we can use the other cell viability tests like annexin stain, and actually, I think theoretically we can do the PI stain as well. Apoptotic priming is the term that we use a lot because we know that the cells in our bodies, it's not like they are alive and then suddenly they are dead. Yes, it takes a while for the cell to die. And we are talking like depending on how close the cell is to its death. Yes, we can talk about the different levels of the priming. We can talk about poorly primed, moderately primed, and highly primed. And with the BH3 profiling, we can actually assess like how close the cells are to the priming, like how sensitive they will gonna be, how prone they will gonna be to die. Yes, and the BH3 profiling basically is a measurement of this balance between the pro-apoptotic and anti-apoptotic events. Yes, so if we have, and basically with this BH3 profiling is rather fast test, yes, so it takes approximately 3 days to do it and get the answer rather rapidly. The basic steps, of course, would be like if we have the cancer biopsy, we isolate the cells, we plate them overnight, and then the next day we can add the drugs. And then we can add the specific BH3 peptides, fix the cells, and stain them with cytochrome C. Yes, and DAPI, and then do the high throughput imaging, and then we can measure like how many cells have lost the cytochrome C. So if the cell has lost the cytochrome C, it means that it's in the process of dying. Yes, so and then we can see we are usually talking about delta priming, so we can see like in DMSO all the cells retain cytochrome C, and when we add the drugs in higher concentrations, these are actual data. The drug actually is DT2216, and the cells are liposarcoma cell line, yes. So we can see like nice dose-dependent decrease of the living cells, yes. So, and we can then explain it, show it as the heat maps and so. Here in Latvia, like how we are, when I came back from USA, I want to implement this BH3 profiling in my lab, in our lab, in Riga Stradins University, and our project is when we are looking at the hypoxia. So one of the tasks in this project, and we are working with the breast cancer cells, with the triple negative breast cancer cells, and we want to know like if, like what strategies are the cancer cells are using to survive the hypoxic environment, and if we know these strategies, and my PhD student Alessia Rogos will going to be talking tomorrow in a young scientist session, so more deeper about that, and please come and see us. She will going to show also the primary data. We have, she's been very great in implementing this BH3 workflow in our lab. So the main questions that we want to know in this research is how the cells in normoxia differ from the cells in hypoxia, yes? So which anti-apoptotic dependencies they will gonna use not to die. And of course, we want to know how to destroy all the cancer cells, yes? And steps forward, international cooperation, as I mentioned, we wrote 2 grant projects. Unfortunately, EP permit didn't go through. 283 projects for 30 places, that happens. Now we are keeping our fingers crossed for the TransCan, which is combination therapies against cancer, which we wrote together with Joan Montero from Spain and Felix Seyfried and Luder Meyer from Ulm. And we're going to know the results of the stage first in October 18th. And in addition, also we are participating in joint action in EU for Health, which is program for the personalized cancer medicine. This is the kind of the soft investment. And in addition, actually yesterday I was moderating the EATRIS kickoff meeting, and I see the benefits also from the working in EATRIS, also making some consortiums and possible calls. And the steps to take forward towards a functional precision medicine, and I'm sorry because this is a very old version of my presentation because I had more slides. But so it's besides the improved material base, I think the very important thing is the communication. And it's the communication not only between the researchers, but between the researchers and clinicians. It's super important. I know that the clinicians are overworked, but we have to talk and we have to work together. But also, I think it's a very, very important thing to talk to the society, to educate the public, not only the public, also the schools and students and young medical people and so on. So I had one more slide where I was kind of wanted to show the great future that we are all moving towards. Of course, we want to increase the treatment efficiency, decrease the costs, decrease the side effects, and of course, decrease the time spent in a hospital, and of course, increase the life expectancy and quality of the patients. That's, I think, all our game, and I think working together, that's the way we can reach it. And I thank you very much for your attention.
Well, thank you, Inese. And just as a matter of time, I'd actually like to welcome all the speakers back to the stage, and we can start a panel discussion. So, yeah, well, thank you all for your presentations. And I think it's really wonderful to see this movement of functional precision medicine sort of moving forward and in some ways now following up from genomic medicine. And I think, you know, all of us are on the same page of that. But, you know, in terms of the thoughts on precision medicine, people are still stuck on genomics, and there's probably still a lot of work that needs to be done in terms of really implementing it. And maybe we can start off with that, and if anybody wants to say more about that.
I think I'm going to start because I'm trying to be, I understand and I see how Tony Letai talks about the precision medicine, and I'm very happy that we have this Society for Functional Precision Medicine, which is worldwide society, and I'm trying to introduce more and more people to this society because it's like, it combines the people from different world. But I think very important thing is just to talk about it. And I know that for me, sometimes I think I'm talking about it all the time. But this is, I think, what you have to do. You have to talk because you meet different people almost every day, every week, and you have to reach it. And I think I'm also trying to be very proactive and through the different scientific podcast, not the podcast, but radio programs like Zināmais Nezināmajā. We are trying to reach the more public as well.
Yeah, I mean, I exactly. I mean, it's so important to raise awareness, and I would just say that the most important aspect here is we need to talk about it in a way that this is the power is combining genomics with drug testing. That's the power there, that, you know, the more information the physician can have, the better he can make decisions, and that's better he or she can make decisions. So what we want to do is, this is a tool that can be added to genomics precision medicine, and let's work together on, you know, generating the data to show the clinical utility of these approaches.
Yeah, and I also think that we have to learn from the sequencing companies. They did really good at marketing. So the oncologists are sold to it, even if they're not convinced that this changes something in the therapeutic regimen. And many of the oncologists I have spoken to had like two arguments. One, sequencing is financed by the public health system largely for many tumor types. So even if they don't expect anything new, they see themselves as service providers, and they don't want their patients to run away to somebody else who offers sequencing. And the other thing that I was hearing a lot of is, your technology with those models is so complex, it takes me more time to explain to the patient, and nobody is paying me for that. So that are basically the two things that run against each other. The great marketing from the sequencing providers versus the time-consuming thing. And I have a total understanding for the oncologists. They want to see their families at the end of the day. They want spare time, and of course they need to be paid for it. But we need to break this dogma to get it into the broader clinic, away from only the university hospitals.
And I think another complaint that I hear also is that we do this type of testing. Everyone does it a little bit differently, and there are sort of no standard practice. But we can argue, actually, in genomics, it was the same way. It started off, and people use different analytical methods. But in the end, it's yes or no. Is that mutation there or not? You know, can we do the same with our different methods in functional precision medicine?
I think this is a very great question, and actually with one of our grants we wanted to address that, and I think we're going to continue submitting those grants for the harmonizing the method, because even the BH3 profiling, it's kind of one method, but yeah, the grant was the one of the steps of the grants was harmonizing this method like all over the 6 labs in the European Union. Yes, and I think even the cell titer glow is like kind of easy method, but I think it also needs the kind of harmonization, standardization.
I guess one thing, going back to your first point, is that the DNA is a solid thing, and it's it lasts and hangs around. Having viable tissue is the issue with the functional precision medicine, and having standardized approaches to ensure that, you know, it is viable and that it matches the starting material is key. I think getting those messages across.
Yeah, I mean, we need to start, you know, making sure our biobanks store viable tissue, right? I mean, that's a big challenge in the field. And I would say the idea of standardizing, we don't have to. I mean, when you're taking this test outside of a research setting, and when you start trying to get CLIA certification, I mean, that alone is showing that your test has all the measures, that it's precise, accurate. I mean, if we are using different tests, but they're CLIA certified, or, I mean, that should be enough for us to... Yeah.
We also went through the pain of getting an ISO accreditation.
There you go.
And you need to validate your setup.
20 replicates, I mean 20 data points for each drug, or 40 data points for each drug.
And you need to show that it's not working only in your hands. So how do you find another lab that does what you are doing to have like a circle experiment where you validate each other's data? So that is just an administrative problem, but we need to tackle this. And I think the Society for Functional Precision Medicine is a good tool to harmonize that, to bring the people together, to organize themselves, and maybe getting some power over the authorities.
Mm-hmm. And I know from Tony's, being in Tony's lab, I think already 2 years they are fighting this FDA. It's like never-ending. It's like never-ending story. Like these regulatory entities, they do not understand how to handle this, and it's. I understand it's a nightmare. But I understand that in Europe we can do it without EMA. We don't need the EMA approval, and
we can do it, but in order for, at least in Germany, for the insurance companies to pay for it, you need some sort of accreditation, and we decided to go for ISO 17025, and that basically shows that everything that leaves your lab is validated, and actually it's a bit also of a loophole, so maybe a little advice for you. Look at this ISO 17025, because then you don't need a CE sign on it, so you can spare that, because by definition things are validated.
I'm sure it's going to be harder in the United States.
Definitely.
Yes, and we're actually at the end of our time, but I just wanted to comment that I think, you know, we agree that it's really important to engage the public, and also, you know, the public, especially patients, have a very strong voice in you know, what the government will allow us to do. So I think it's important that we reach out to that group as well. And
I agree. I just want to add that, you know, in my clinical trials, we give the results back to the doctors, and I've witnessed situations where the patients don't even know about the results. So when we're CLIA certified, and when the patient is empowered and knows that there's more data that can be used to guide treatment, I think that's how we can empower the patient and give that information. It's important.
any other last comments?
Just the last thing about the information. I was just thinking, I remember this year, at the beginning of this year, I invited the patient organization to my lab to come, and I gave them the tour around the lab. I explained them what's the functional precision medicine, what it can do, in trying to do it like in very easy words and like very relaxed environment and so on. So I think we have to be proactive towards the patients as well. We cannot expect patients to come to us. We have to go to them. And also another thing that I've been very happy and lucky, I already gave the talk in a children's hospital 2 weeks ago, and I hope that soon I'm going to give the talk in other hospital in Riga, Riga Eastern Hospital. I think we need to inform the clinicians about these possibilities more and more as much as we can.
If you are interested in a nice format for promoting science, we're doing something with a patient organization. We call it a pint of science. So you sit in a pub, you have beer, and you talk about science. So that's a perfect mixture.
Might have started in Ireland.
Yes.
All right. Well, I'd like to thank all the speakers and the audience for staying during the session, and very welcome now to go get a cup of coffee and then take a break. Thank you.